Yo, 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 welcome to Unit 6.1, Gene Expression. Today, we're going to cover Topic 1, Proving DNA is the Genetic Material. In the early 1900s, scientists struggled to identify which molecule contains the genes that are involved in inheritance. Two scientists contributed to that, and it narrowed down to the identity of the genetic material to two types of molecules found in chromosomes, proteins and DNA. Griffith experimented with a bacterium that causes pneumonia in mammals. He used two strains, one that's pathogenic, meaning disease-causing, and another strain non-pathogenic or harmless. He found that when the pathogenic bacteria were killed with heat and then mixed with the living non-pathogenic, the living cells became pathogenic. Whoa, whoa, whoa. This phenomenon was called transformation. Not that he knew of that principle at the time, but he couldn't figure out or narrow it down to which part of the foreign DNA is causing the transformation. It could have been carbohydrates, proteins, or nucleic acids. In Griffith's experiment, the bacteria, the smooth strain, are the pathogenic ones because they contain a sugar capsule that protects them from an animal's immune system. The rough strain lack that capsule and are non-pathogenic. And so as you can see, when the heat-killed S cells are injected into the mouse is healthy. But when it is mixed with the living rough capsule, cells, it became pathogenic. And so living S cells were found in the blood sample of the mouse that died. Taking the idea from Griffiths, three other scientists decided to test this idea out of the transforming principle. In their experiment, they isolated different solutions containing the heat-killed S bacteria cells and examined which solutions transformation still occurred with the R strain bacteria. Only the solutions with the S strain DNA still intact led to the transformation of the R strain bacteria. But the scientific community was still very skeptical of these findings, as they should be. Again, testing and testing and testing. Many scientists still supported the theory that proteins were the genetic material. So this experiment was described on the previous slide, but now visually speaking. We have a sample of heat-killed S cells and lipids and carbohydrates are removed from that solution. That solution is then separated into three samples. In the first sample, we see that proteinases, which breaks down proteins, were added to that sample, and then R cells added. Well, transformation occurred because RNA and DNA is still intact. Let's see what happens with the second one. The second sample, the scientists add ribonuclease, which eliminated RNA. So no RNA should be in that solution. The R cells are added and boom, transformation occurred. So again, the commonality is DNA is in both. Last but not least, let's see what happens when DNA is gotten rid of by adding deoxyribonuclease. So when that is added, again, the R cells were added, no transformation occurred. So the only logical conclusion is transformation cannot occur unless DNA is present. Whoa, whoa, whoa. So further support for DNA came from Hershey and Chase's experiment. Their experiment showed that DNA was genetic material of a bacteriophage known as T2. So what is a bacteriophage or a phage? It's just a virus that infects bacteria. So T2 viruses are composed of mostly DNA and protein. When they infect a bacteria cell, T2 reprograms its hosts to produce viruses until the cell ruptures, releasing all those viruses. Their experiment involved radioactively labeling the proteins and specifically sulfur, and then DNA, specifically the phosphorus, of T2 viruses and seeing which of the two were found in the bacteria cells that were infected by the virus. The results showed that viruses transferred their DNA, not protein, in your face. And, you know, DNA is the winner. So again, this is the experiment. So labeled phages, specifically with the sulfur, infect cells, agitation frees outside phage parts from the cell, and then centrifuging it allows the free phage parts to remain in the liquid. And so what was found? Radioactive phages protein was found in the liquid, and then not within the bacteria. But when we see with phosphorus, the label for the phage's DNA, same process occurred, centrifuging occurred, and guess what, y'all? The radioactive phage DNA was found inside the pellet, meaning again, the DNA is what was loaded inside that bacteria. It's been decided. So, DNA is the genetic material. Now, diving into topic 2, the structure of DNA. So, we went over the structure of DNA during the first unit. Most scientists were convinced that DNA was genetic material by the 1950s, and it is known that DNA is a polymer of nucleotides, which again consists of nitrogenous base, 5-carbon sugar, and a phosphate group. And the 3D structure is still unknown at that time, and we'll cover a little history. But let's move on to that structure specifically with deoxyribose. So the name for that 5-carbon sugar, as previously mentioned, is deoxyribose, and that's specific to DNA. Now we have that 1' to 5' numbering system, as we can see here, and that is numbering the carbon atoms in the pentose sugar. So the nitrogenous base is bound to the 1' carbon, whereas the phosphate group is bound to the 5' carbon. Another interesting fact is the 3' carbon has an OH group attached to it. That will be important later on. So there are two families of nitrogenous bases that you guys need to remember. The first one is pyrimidines, bases with one ring, and includes cytosine, thymine, and uracil. So one way to remember pyrimidines being one ring, and that it is, including cytosine, thymine, and uracil, is to say cut pi. Cut is one word, and then pi stands for that pyrimidine. Cut makes up cytosine, uracil, and thymine. Then we have the other family, which is called purines. And so they are bases with two rings and includes adenine and guanine. So we can remember it by saying pure as gold. As gold is two words for two rings, and then the initial letter there, A and G, as gold, adenine and guanine. Boom, there you have it. So nucleotides are joined together by a series of dehydration reactions. And remember, dehydration simply means we're losing that water to join things together. Okay? So covalent bonds are formed between the 3 carbon and then the phosphate group of the next nucleotide. This link is called the phosphodiester linkage. And you guys can see it here. Boom, the OH with this phosphate here on another nucleotide. Again, dehydration, we lose water to form that connection here. Polynucleotides have two ends, a 3' hydroxyl end and a 5' phosphate end. During DNA and RNA synthesis, nucleotides are added to the 3' end of a growing strand. So now that we got the basic structure of DNA out of the way, Franklin and Wilkins used X-ray crystallography to study the 3D structure of DNA. So these images comes up with spots and smudges created by the diffraction of X-rays passing through molecules. So mathematical equations translate the patterns into information about the 3D shape of the molecules itself. Franklin had concluded that there are two outer sugar phosphate backbones with the nitrogenous base paired in the interior, forming a helical structure of sorts based off of this image here. At the same time, these two guys were trying to uncover the structure of DNA as well. Watson used Franklin's X-ray images to deduce that the DNA was helical, as well as able to calculate the width and spacing of the nitrogenous bases. The width suggests that the DNA molecule was made up of two strands forming a double helix. So Watson and Crick developed the final model of DNA in 1953. At first, Watson and Crick thought that the bases paired like A with A and so on and so forth, but such pairing didn't result in a uniform width. Instead, pairing a purine with a pyrimidine resulted in a uniform width consistent with the X-rays. Pretty cool, right? Math. Boom. So Watson and Crick determined that adenine pairs only with thymine and that guanine pairs only with cytosine. Matching their findings, Chargaff again discovered that an organism's DNA-based composition of adenine always equals thymine, and same for cytosine and guanine. So this was Chargaff's rule. Again, this simply means the same percentage of A and T, and then the same percentage of G and C can be found in an organism with a double helix DNA and double helix RNA, if we're looking at RNA. So, reviewing DNA's 3D structure. First, DNA is a double-stranded molecule. Both strands are made up of a backbone that's composed of alternating sugar phosphate molecules. The two strands twist around each other, forming a double helix, and hydrogen bonds are holding the nitrogenous bases together. There's complementary base pairing between the DNA strands. G and C will always be paired together, forming three hydrogen bonds, whereas adenine and thymine, only in DNA, would form two hydrogen bonds. So as previously mentioned, the DNA strand has two ends, one that's considered to be the 5 prime, that carbon there attached to a phosphate group, and then this carbon here attached to that OH group. Okay, and so we have that direction. So we know it's a 5 prime end because of this phosphate, and we know this is a 3 prime end because of the hydroxyl group. And if one goes from 5 to 3, then 5 to 3 would run up on the other strand. We can see the backwardness of that DNA strand there. So this is called antiparallel. They are oriented in opposite directions. The DNA inside prokaryotes is organized into a single circular chromosome as seen here. And then the eukaryotes have a linear but compressed chromosome. Now, bacteria, cells, viruses, and some eukaryotes contain plasmids. But what are plasmids? Plasmids are small circular double-stranded DNA that are distinct from a cell's chromosomal DNA. And these genes in plasmids provide bacteria with genetic advantages such as antibiotic resistance. So you can see that it's definitely different from the bacterial DNA, making all the other functions. These plasmids usually provide additional functions. Okay, that's the difference between prokaryotes and eukaryotes. And it's that time again. Have a good one.